This is Dave Monk, Prairie Monk, WEFT, Champaign 90.1, your FM dial.
And Dave on the board.
And, yeah, have a few rants today.
Happy Father's Day, by the way, to everyone out there.
Yes, we appreciate fathers and what they do and the time it takes to work with family and children. I, again, pulled things out of the garbage can, and this time it was a bar stool. And the bar stool had a structurally quite good, the seat had disconvolvulated a little bit, but I took it around to 4 or 5 different agencies. No one would take the bar stool. The problem is that we don't have any repair organization. We have people who will generously give their help to putting in a screw here or something simple. But if people like PACA, the Preservation and Conservation Association of Champaign County, is interested in some broader issues. One of them is that we might want to encourage employment of people who know how to repair. That doesn't mean it's just people standing there and accepting material. It means that you have to have enough knowledge and be sort of jack of all trades to put something together. And also know how to disconvolvulate some things. For instance, if you have a motor, the motor is heavy in copper, and that means that it should be in a different category for salvage than if you put it with plastic, then it goes to appliances, and you don't get any salvage value for it. The salvage value is not all that great. It's not enough to prevent people from saying, well, we don't want a compression wood. We'll go to Indonesia or the Amazon and take down a few more trees. So I took apart several shelves. They were put into the garbage bin whole, so they took up a huge amount of space, and I broke them down carefully, and they each were about 3 feet long and similar size, and I thought that people who want to stack books could perhaps use these. But we're not in that frame of mind yet. We either go to supermarkets and buy the same thing new because it's new, or we don't think about recycling that. So bar stool I still have if somebody is interested in the bar stool. This structure of that chair is quite good. The fake leather on it is good, and it just needs a little bit of attention for the seat. The compressed wood I probably will have to put back in the trash can and go off to Clinton or Danville for a 35-mile trip and sit in the landfill, and it will have some glue in it because it's chipped material that is glued together to make compression board. So that we have to still think about the fact that we don't have commercial recycling in downtown Champaign. Other cities in the country do it very well. I go to the idea store and they do that sort of thing well. We had Kim Campbell here yesterday, and he was helping me. Ron Sleater is aware that at this stage the teasel is getting to be high and needs to be herbicided, spot herbicided. And the clover that the Department of Transportation plants on top of our prairies sometimes is very dangerous for us. It's a weed, it's a farm weed. It's been a very useful crop, but we put together several rows of experimental herbiciding machines. The thought that Ron had was that you have a person on one end and a person on the other end of the rope, and you dangle herbicided fabric down, and you can run it across the height of the sweet clover. And the sweet clover grows faster than any prairie plant, so you usually would hit the clover with a systemic herbicide. We as conservationists, you might think that we might not use herbicide, but sometimes we're in a situation where we have no other alternative. The best way of doing this is to take the plant when it's wet, when the soil is damp, and pull it out. But then again, we're in conflict with people who don't have much flowering material for bees, and they would like these. And if you go along the road, all the roadsides seem to have somewhere this mysterious bulk of seed that gets planted. It's sort of felt the sweet clover is a fertilizer, natural fertilizer, in that it makes its own nitrogen and can fertilize as legumes do, has rhizobacteria on the roots. But really, when you're adjacent to the only natural areas that we have, then that's pretty cruel. But it comes in big sections. You can see it live at the moment. There's two varieties. One is yellow, one is white. The white one hasn't come yet. The yellow one is out in bloom at the present time. So we were intending to wait till the rain finishes, because no point in herbiciding and then having the rain fall. So we would walk. We've tried two different things. One is with a rope, and the other one is with a pole. The pole being a little firmer, and the thought is, do we have varying lengths of material? But the idea store gave us the material, or we bought the material there, and they had happened to be from a sewing people where they'd taken the cuffs off of jeans, and so this meant that we could put the material on a pole and staple it together, and we'll douse that with herbicide and walk through a plot of yellow clover that we could not possibly pull with our hands or with our backs or what have you. I also wanted to say that there was a program yesterday on WIL that People's Pharmacy, and we talk about zoonosis here, and zoonosis is a study of how diseases float from animal to animal and across to humans sometimes, and so that program, Graydon and Graydon, was dealing with a new bacterium. It's probably not new, but it's newly discovered. It's Bartonella, and it's hard to define. It's a disease that actually gets into your blood cells, and it's hard to detect. But the symptoms are that it may be associated with arthritis. It has to do with your vascular system. It'll narrow your vascular system some, so then that has to do with your nerves, because your nerves need blood vessels. And so you can be walking a little strangely, but it has a difference from something like Lyme disease. And it was pointing out that as you get more sophisticated, the tests that you get at a doctor's laboratory are fairly simplistic. If you're going to look at people that have Bartonella, you probably don't have a standard test that you can use. And so the researchers, and these were very good researchers, I thought, and one of the things that's very helpful is that they can express themselves pretty well and tell you what's going on. So the researcher that I was listening to had been dealing with Lyme disease, because that's another disease that you don't exactly know what the symptoms are and how they're going to work out until you've done some research. And to do research, you really need money to do research. And often, if you have a vague disease, it doesn't get money until it really hits. It's like AIDS was very vague disease until it really started to hit people, and then you get the money to do it. So you have to design your research on a very small budget. And in this case, they know about Bartonella's disease. It's called cat scratch disease. And so they targeted the groups that would be likely to be associated with fleas and sucking insects or what have you.
She's here.
Okay.
Keep going. Come on in.
So anyway, let me just finish on that. It just was intriguing to me to know the complexity of this, and it's a warning to people. We're just talking about herbiciding, and we're walking through Prairie, and that means we get ticks. And there are other people veterinarians who save dogs, and the dogs may be in a pound where they're full of fleas and other things like that. And then there were cats. And so the researchers targeted those audiences, or those people, those professions, and started to find out things about Bartonella's disease that eventually will allow them to define what it is. If it's so small that you can hardly find it, then you have to grow it in an incubator till it gets to be known enough that you can test it. Then you have to find out a test that can be handled by doctors, and then doctors have to know about it, and that's all quite complex. And you have to have an association between a researcher and the person who's in the field, who's thinking, these people all have rheumatism, but it's not like rheumatism that's coming from rheumatism. So how do you work the scientist that's the researcher together with the people out there? And then we have to turn around and suggest to people like ourselves who walk through the prairie that if you have ticks on you, you better just check that out. And then I'm wanting to do herbiciding, and I'm wanting to get volunteers to help me do that. And so when I tell you that there are ticks out there, you might not want to come and help me. But how do we do this without being in contact with the animals that are around? If you're in a native situation, you might have little flies and fleas and lesser fleas and what have you that are going to be part of your life. And how do you work with malaria and things like that? So that's enough for now. Michelle Dwayne has come in. She's here.
I've got a question.
Hello.
So he was just talking about ticks. So Michelle, you must know about ticks because you work with bees. There's almost no difference. How much? My question is, how much of what you know about bees translates to some other insect, some other thing like a tick?
As for part of my PhD, I have to take a bunch of different classes about insects, and ticks are not actually insects. So they're arachnids, but we do learn about ticks and tick-borne diseases in our entomology classes because arachnids are closely related and important disease vectors. So we do learn. I do have to learn about them for my PhD, even though I don't really study them.
So it comes under the term zoonosis in the veterinary medicine area. Zoonosis being where you have someone that's looking at human diseases and animal diseases and their transmission and who are the vectors and what have you. But, okay. Well, we have Michelle on for a second time, I think.
I think this is my second time.
So we get together because she asked me to do a field trip for pollinators. There's Pollinator Week coming up.
Yes.
And so we got together and we've done one or two pollinator trips. In fact, we filmed one. After we'd done the trip, we had a videographer come and do it again just to check it out. And this is going to be run on the 23rd, which is next week, I think.
Yes.
And Sunday it's at 11 o'clock? No, 10 o'clock.
10 o'clock.
10 o'clock. Tell me a little bit, are you going to have a photographer there like you usually do?
Yes. So we're doing David is going to do a nature walk at Meadowbrook Park from 10 to noon. And then from noon to 1, Alex Wild, who's a local insect photographer, but his photography, his entomology photography is among the best in the world and is in textbooks, and he's an amazing photographer, and he'll be giving a photo workshop at Meadowbrook right after the nature walk.
Yes, and he is very good, and he suggests to you what sort of cameras you might use, and sometimes very simple things that you can do to put a lens over your lens so that you can get the head of an ant, so the ant looks more normal. You don't have the nose sticking out and the back way back there. He knows how to light things. And so I had last night Kim Campbell here, and he has just bought a Canon camera on Alex's recommendation. So things come out of that program, and sometimes Alex is also an entomologist, and he comes and points to little insects that you would never think to see because they're not obvious. They might be feeding on the guttation pores that are on a leaf.
Yes, Alex also has his PhD in entomology and teaches classes at the university, so he's also an excellent entomologist and photographer. So
yes, so
wonderful combination.
Yes, so that will be there, and he will sometimes come into the Prairie too, so that's helpful. So that will be at Meadowbrook Park, and we'll take you on some trails that aren't 12 feet wide and suitable for a tandem truck to run around. Meadowbrook Park has that sort of that sort of trail, and that's where runners run and the like. But there's a little trail that is a mud trail down the Ombra River. It's the head of the Ombra River, and you have to think about Yankee Ridge High and Meadowbrook has this little creek that runs into the Ombra, and then the nearby Pasota Moraine is to the west, and it runs into the Ombra. The Yankee Ridge is more where the glacier used to sit and dribble in to the backside of the Pasota Moraine. The creeks are not as steep or vigorous. So we'll be on the Yankee Ridge Moraine, and we'll talk to you about some of those sorts of things and how ecology sorts itself out. And then if you plant plants on Meadowbrook Park, you have to figure out what you want to do, because this was a cornfield, and how do you simulate what you have in 1830 when we settled here. So let me ask you to talk, because you have been doing exciting things in Panama and Mexico. So tell us a little bit about what you're doing.
So I just got back from a 3-week trip across Central America to study bumblebees, and so I was in Panama for a while, sort of vacation, but also just to kind of survey what kind of bumblebee diversity is there. And then I went to Mexico, the southern part of Mexico in Chiapas, and went to a museum that I've been working with there, and was pulling out specimens of bumblebees from across Mexico to do population genetics on one of the most common species throughout Mexico that has really interesting colors, really interesting habitat that it lives in, and it's a really important pollinator for that region of the world.
Is it healthy?
The bee, so
Yes.
that's part of this project is what we're trying to find out. So my part of the project is I'm going to look at the genetics of the bee and see does it have a lot of genetic diversity, which is a good thing? Does it have low genetic diversity, which might indicate some inbreeding? Does it have really fragmented populations, which might be a result of habitat fragmentation? But another part of this project, that's another really large part, is every bumblebee specimen that they collect, they're pulling the digestive tract out of, and they're looking for pathogen infection. So looking for primarily two major pathogens in bumblebees. One is nosema, which you might have heard about in honeybees, because it also is a serious infection that can occur in honeybees. And that's fungus. It's sort of like a fungus. It's actually called a microsporidium, and it gives the bees like a really bad infection, and it can be really bad for colonies. They get really sick. It makes the colony really dirty. And it can be a really big problem in bumblebees. It can be a big problem in honeybees. And the other one that they're looking at is another pathogen called Crithidia. And I don't know too much about that, but it's another major bumblebee pathogen. And we're just starting that work. So we're just now trying to figure out how that's affecting the conservation of bumblebees in Mexico.
And just tell the population that's listening, or the listening audience, just a little bit more about bumblebees. They're very different from a regular bee.
Yes, they are. They have a very different biology from honeybees. So you may know that with honeybees, they live in these boxes that we put them in, and you can have tens of thousands of individuals in a single colony, and you have one queen, and she is the mother of all of these workers and drones, and you have tens of thousands of workers. But in a bumblebee colony, it's quite a bit smaller, and they live underground. So a bumblebee queen will emerge in the spring. She finds before spring, at the end of fall, she finds a little tiny place called a hibernacula to hide during winter, and she overwinters there. She kind of hibernates, and she has already been mated at the end of the fall. So as soon as she comes out, she's ready to lay eggs, and she needs to find a place to raise all of her daughters. So she flies around, and she finds a nice nest, and usually she'll find something like an old rodent nest, or and they can even use hair that's left around in the colony to insulate it. So they usually find something like a rodent nest. Some species will nest underneath grass piles. And when she finds a really nice place to raise her young, she'll then start collecting nectar and pollen, and she makes this giant honey pot, just one single honey pot, and she'll fill it with nectar and pollen, and just fill it, so that she has enough food to feed on while she incubates her young. So she'll fill up this big pot, and then she'll lay her eggs, and then she plops down on top of those eggs and just eats out of that pot for a couple of weeks while she incubates them.
So
yeah.
laying eggs, it's an intentional thing for her. It's not like in human beings where, maybe we're having a baby now. Oops, nope, false alarm.
No,
we'll go back. And she says, okay, and now I shall lay my eggs. And there she goes.
Yes, they have such remarkable control over reproduction. They can even control when they're going to make males and females. So a male, another thing about bees that's really interesting and completely foreign to human ideas of reproduction is that an unfertilized egg will become a male, and they're haploid, which means they only have one copy of their genome. But a fertilized egg will become a female, and she has two copies of her genome. It's just crazy how they reproduce.
That's very helpful. But you might say how they're useful because pollinators.
Oh,
this is
right.
Pollinator wheat, and they are very good pollinators.
Yes, they're amazing pollinators. So they're really good pollinators of blackberries and blueberries and cranberries, and the crop that they're the best pollinators for are tomatoes, because tomatoes have what are called poricidal anthers, and they need to be buzz pollinated. So they have this poricidal anthers. And so what that basically means is they've got kind of this capsule with holes in it, and it needs to be shaken up so all the pollen can come out.
And bumblebees are bigger.
Bumblebees are bigger, and what they can actually do is they have these really powerful flight muscles, and they can detach those from their wings and buzz them separately, and that will shake all the pollen out of these poricidal anthers. And so they're really good pollinators of tomatoes. So in greenhouses, if you're growing tomatoes in a greenhouse, you can order bumblebees from a colony that raises them in the lab, or from a company that raises them in the lab, and then they'll send them to your greenhouse, and then they'll pollinate your tomatoes for you. And I actually found out recently that one of the tomato growers that we've kind of been camped next to at the Urbana Market at the Square, I was talking to her a lot last year about using bumblebees in her greenhouses, and she was telling me that, if she was at the time doing it by hand, so they have a machine that you can buy you just basically vibrate the flowers so that so they can pollinate them. And she said if they don't do it at the right time, they get these little growths and bumps, and then they can't sell the tomatoes. And she had to use a lot of man labor. And I said, oh, well, you should get bumblebees because they know exactly when to do it. And they're really the best at it. They have been evolved to pollinate these tomatoes. And so I found out this year that she has actually started using bumblebees in her greenhouse. So
Was it successful? You
I don't know.
The season hasn't come yet.
So I've been out of the country, so I haven't been able to talk to her in person, so I don't know if it's working out that well for her, but I've heard from word of mouth that she's really happy with them. So
are you going to be a geneticist that's looking for diseases? You're talking about the genetics here, haploid and diploid organisms. So yes, tell us what your aim is in doing your thesis. Will that continue to a job that has that sort of thrust?
So I really like population genetics, and I think it would be really so for my PhD right now, I'm just looking at the genetics of the bees themselves and seeing how the mountain ranges of Mexico have shaped their genetic structure throughout the country.
So a bumblebee that's high in the mountain will be different from one that's down low.
Right. Well, that's my hypothesis. So the one I'm looking at right now, its name is Bombus afipiatus. It doesn't have a common name, but it's kind of a mouthful. But it lives really high in the mountains. So what I've already found with some of the data I've collected so far is that in Mexico, you've got a very distinct genetic group. And then in the southern part of Mexico and in Honduras, you've got another very, very different genetic group. And then in Costa Rica, you've got this completely separate group too. So, and these all correlate to the elevation. So they all correlate to the different mountain ranges that go throughout Mexico and Central America.
So I have problems because in Mexico City area, you have Popocatépetl, or I don't know how to pronounce it, but there's a very high mountain.
Yeah.
And even Mexico is on a plateau. So when you go up there, even in summer, it's still snow.
Yeah.
So do your bumblebees work in snow?
Yes, bumblebees actually are really good at tolerating cold temperatures. So if there are in some areas there are flowers that will bloom while there's still snow on the ground, and the bumblebees are capable of utilizing that because they can actually generate their own body heat. So so I talked about how they can decouple their wing muscles from their wings to vibrate the flowers. They can also do that to generate body heat.
So they can shiver.
Yeah.
Wow.
They're really cool.
but population generates in my mind statistics, and fairly huge statistics. You're trying to get enough thousands of insects to
Yes.
give you an idea of what the norm is, and the norm will be a bell-shaped curve. It won't be monolithic all the same.
Yeah, it depends on it depends on how the environment has shaped the genetic structure of the bees in that area. I really don't know what I'll find, but I'm pretty excited about all the bees I brought back with me. So
You bring back live bees?
no, so the bees I brought back are from the museum collections in Mexico, and those are all I bring back are legs from these museum specimens. So all the bees stay there, and I just kind of pull a leg off, and that's enough for me to get DNA from the specimen.
So tell us about your lab experience. When you put the leg, do you put it in a little file or?
Yeah, I have a really tiny little tube that I put the leg in when I pull it off the bee in the collection.
Uh-huh.
And then I use a bunch of different enzymes and solutions to break down the tissues inside the leg
You get
to
the
get
RNA
the
and
DNA.
DNA separated. Then you have to work on that.
Yeah. Mm-hmm.
Okay. And so how many legs do you have to have a sample to be able to work with? This is almost nanotechnology.
Yeah. They're very small little pieces, and actually, I have about 750 bee samples from Mexico, and 300 of them are the whole leg, and the rest of them, I pulled the leg off and clipped it in half, and so I'm only using half of a leg, and then the museum that I'm working with keeps the other half of the leg. So I'm working with very small amounts of tissue.
So you have small amounts of tissue. Do you have to grow your DNA to the point that you have enough to be able to test?
No, not with what I'm doing.
Uh-huh.
Mm-hmm.
Okay.
So I'm glad I don't have to do that.
So you have to have microbiological techniques because
Yep.
if you're
Mm-hmm.
not very careful, your leg will get infected with what's in the air around you.
Yeah, so it's not as much of a problem when I'm pulling the leg off of the bee because when it's sitting in a box in a museum, it's really hard to. It's going to come in contact with the air in the museum, all the other bees in the box. In one of the boxes, we found some beetles that had ended up eating some of the specimens, so there's that stuff flying around in there. But most of the DNA that I'm going to get out of that is going to be from the bumblebee, so it's more about concentration than anything else. But I have to be very, very sterile with my extractions and with my... So once I remove the DNA, I have a certain piece of the DNA that I want to look at, and so then I will amplify that millions of times with a separate reaction. So I have to be really sterile with the DNA extraction and the amplification, but not. It's kind of unavoidable in the beginning parts when I pull the legs off.
So can you tell people how you check for the DNA? Is that?
So it's a little complicated to explain,
But you've been here before, so
Yeah.
we know basically what you do.
Yeah.
So this is taking it to a little more sophisticated
Mm-hmm.
level. People out there need to know this sort
Yeah.
of stuff. There's a disconnect between scientists and the public.
Yeah, so I study bumblebees, but a lot. I would say it's. I really like telling people that I'm a scientist and, I study bumblebees and I go to these exact places to collect them. But I'd say 90% of the time I'm working on a computer or I'm in the lab pipetting is taking really tiny amounts of liquid and moving them around in tubes. So that's traveling around and collecting bumblebees in Mexico is not is not what I'm doing all the time. It's what I do about 10% of the time.
Yes. So, do you detect any collapse, colony collapse sort of thing in Mexico?
So that's what we're looking at right now with pulling out the guts and looking at that. And I was at a workshop last year in Mexico where we decided how we were going to go about with this project, because so this project is part of the bumblebee conservation, bumblebee specialist group for the International Union for the Conservation of Nature. I think I'm getting that right. IUCN. I think I have that, all the words in that acronym right. So we're a group that's specifically trying to map out the distribution of bumblebees across the whole world, find out where they live, how many of them are there, are they really rare, are they really common, and then also look and see if they're endangered because they're really rare or because they have really high pathogen loads and things like that.
So we've heard a lot lately about Europe. And Europe cancelling nicotinoids that are herbicide. Is it herbicide? No, it's insecticide.
Insecticide, yeah, neonicotinoids.
So is Europe having problems with its bumblebees? Or is this a target for the Italian bee?
Yes, actually, there's been a really extensive research into bumblebees in Europe, and there are quite a few species that have gone extinct or are endangered in Europe. But at this point, we can't say whether or not it's because of pesticides or habitat fragmentation, or most likely it's a combination of a lot of different factors.
There's a lot of ifs, but it seems to me that the politicians are starting to eliminate the nicotinamides.
Yeah. So they did ban neonicotinoids in Europe recently.
So are we having the same problem here?
The research so far, there's only been a few studies into how neonicotinoids are affecting bee populations in the United States, so it's there are a few that have said that have shown that they do seem to affect bees, but I would say it's not conclusive yet, as it's definite. I think it's safe to say that neonicotinoids are not the only reason for bee decline.
Oh, yes. So you may have 7 different diseases,
right?
And what it does is actually stress the bee to the point that the bee can't do its work.
Right. For me personally, I think we've been focusing a lot on neonicotinoids because there's been a lot of focus in the press, but all you really have to do is look at the geography of Illinois and see that there's nearly no food left for them to eat and hardly anywhere for them to live anymore.
So I wonder, as I go into a soybean field, now it does have a flower. If you go to corn, it doesn't have, it's wind pollinated, not bee pollinated. But do soybean fields provide a base for bees?
Yes, but honeybees will feed on soybean. So,
what intrigues me is we were just talking about zoonosis, the very complex nature of the biochemistry that you have to deal with. We're talking about with humans with brains, but you're talking about bees that also have brains, and absolute minor amount of a biochemical can change things. And so I imagine we know very little about a bee's brain.
We actually know a whole lot about a bee's brain.
Oh, okay. tell us about a bee's brain.
And a lot of the research that's been done on bee brains has been done here at the University of Illinois by Gene Robinson and his lab. So we actually know a lot about what sorts of signaling pathways occur in bee brains to cause certain behaviors. And so that's a lot of what Gene Robinson's lab is looking at currently too, is if you give a bee this compound, how does that change its behavior?
Yes, it seems like that's part of the argument about nicotinoids,
right?
Is that if you give them nicotinoids, the bees go out and they don't come back. They've lost their flight pattern.
Right. Yeah. So it does have it does seem to have a neurological effect in bees.
Okay.
Which a lot of pesticides do. That's their main mode of action, is to interfere with neurological pathways, and that's why some pesticides are so effective, is because they interfere with their neurological pathway in an insect, and then they kill them.
But it gets everything.
Yeah. But sometimes there's non-target effects, so they affect things that they're not supposed to, like. So they kill the bugs that are feeding on the crops, but they also kill other things that
You want
they're
around.
not supposed to.
Yes. Yes, and there's a tendency to pick up an idea that's very simplistic when it's not very it's simplistic.
How could they do that? I mean, you would think that a substance that's sufficient to have a neurologically effect on, say, a dog would have the same effect on a person too. So you can't have, it couldn't be specific enough to knock out the beetle that's the bad thing and leave the bumblebee, the honeybee, in safely.
Yeah, some pesticides or some pest management vectors are specific. So things like BT, Bacillus thuringiensis toxin, so you have the transgenic BT corn, that is actually supposed to only really affect lepidopterans like butterflies and moths. That's actually really specific. And as far as we know, it really only affects lepidopterans. But other things, a lot of pesticides kind of go on the market without completely understanding their non-target effects on what they're not supposed to be working on in the first place. So, but normally they'll go through a mammalian trial to make sure that they're not harmful for people. Right. But they don't really explore too much whether or not they're going to harm the insects they're not supposed to.
There's not a quarantine period often. You do it fairly quickly. You don't do it in the quarantine for 10 years or something like that.
Is this true in other parts of the world as well? Like in Europe, could you get away with putting something in the market? Hey, look, it kills this kind of a bug. Oops, there go all of the honeybees too.
I don't know too much about the regulation of pesticides in other countries, but I imagine they probably have a similar process. I don't really know too much.
We were talking about a veterinarian disease that affects your vascular system because neurological things need blood too. So in insects you have a hemocoel, not a blood, a human blood.
Yeah.
So is there a situation where your nervous system in the bee is affected by the hemocoel? So do you study the hemocoel as well?
Yeah. So so the blood cells will anything that gets into the blood will feed into all the other parts of the body as well. So there's lots of, everything in the body is connected. And within insect system, their breathing is very different from ours. They have a tracheal system where it's oxygen straight into the body. And so they have these holes called spiracles where oxygen will go straight into the body and it feeds directly into the blood system. So it's sort of like a lung, but very, very different.
So I have a thought. If you had a high school student working with you and you're talking about pulling out the guts or pulling out the nervous system, how would you go about teaching the high school student to take out a nervous system out of a bug or a bee? Or
so I don't have much experience with working on the nervous system of a bee, but I have been pulling out hundreds of guts out of bees in my lab on a project that's not for my dissertation, but is a separate project we're working on in my lab where we're doing something called metagenomics, where we're using genomic sequencing techniques, so techniques you'd use to sequence the whole genome of an organism to look at the complete microbiota of the inside of a bumblebee. So we've been looking at species from all over the world, and I've been pulling all their guts out so we can look at them. And with a bumblebee, it's actually fairly easy to pull the gut out. And I imagine it would be as simple for the rest of the insects, too, because they have a very simple digestive tract. They have a foregut, a midgut and a hindgut, and it's all very connected. So how I pull it out is I just pull on the last segment of the abdomen, and I pull that off, and then usually the whole digestive tract just comes right out.
Uh-huh.
And usually, well, it's kind of gruesome, but usually I'll cut off the head as well to make sure that it's not still connected to the head and doesn't rip apart. But and what I think is really interesting, but maybe kind of gross, is sometimes when I pull the digestive tract out of the bumblebee, they've got something called a crop where they'll store honey that they bring back to the colony. And sometimes, depending on when you caught the bee, you'll pull its crop out, and it's just full of honey. It's just like this little bubble of honey when you pull it out. But
Well, that reminds me that honey has more in it than just sugars.
yes, honey has lots of things in it.
And it's very important for the bees themselves.
Yes.
As well as for humans that might be taking it. So how does the educated high school student find out about these sorts of things? How do we take biology to the level that you're talking about so people understand it?
Well, I'm hoping that with National Pollinator Week, we can educate people a little more about the biology and importance of bees. And we do a lot of outreach with in the Department of Entomology to teach people more about the biology of insects. And we do a lot of outreach with high school students as well. So we go into the schools and we teach them about what we're working on. And so we have a lot of outreach programs in the School of Integrative Biology as a whole to try to teach the community about what we're doing.
And one of the things that people have to do is you have to do mathematics and
Yes, I don't really enjoy it that much, but I'm kind of not very good at math.
it hasn't been something that female people have been encouraged to do, even though female brains do it much better than males often. So you'd have to encourage your high school students to look at math.
Yes,
So
I really wish I had not been as afraid of it as I had been in high school and college, and I didn't really understand how much of it I wouldn't have to need to do when I became a biologist. I just thought I'd be, hiking through the jungle and collecting bees, and I never realized I would need to know how to do any sort of computer programming or do that much mathematics or know really complicated algorithms for all these different theoretical models that I have to use in my work.
So I'm thinking of blue water, and if you have enough bumblebees collected throughout the world, will you be working with something like a supercomputer to pull out different pieces of information?
Yes, so we actually have we don't have a supercomputer in our lab, but we have a computer with 8 processors that runs all of our really high input computer analyses that we have to. So there are a lot of when you're studying the genetics of organisms in anything from population genetics to looking at their genome, you now have to use very fast computers to be able to run these really large analyses to look at the data. So we have a lot of facilities on campus that allow us to run these really intensive analyses.
So how do you possibly get a job elsewhere in the world with that sort of need?
So there are a lot of listservs that I've signed up for looking for jobs, and there are lots of jobs. There's so many jobs now in biology looking for specifically for bioinformaticians. So that's someone who has a lot of experience in computer programming and statistics. And there's lots of jobs because we have a lot of biologists now who don't have that training, but they need it. So they're hiring lots of computer programmers and biology programs now to help
people my age
people.
don't have those skills, folks.
Yeah.
They have ideas, but they don't have those sorts of skills.
Right.
And what I want to say is that mathematics is not only a help, but it has to be to the point where you enjoy it, where you get your jollies out of doing it. There's a spirit and a passion here that comes with it, and that's rather hard to kick into.
Yeah, I have a great appreciation for mathematics. I just don't. I don't really like doing it, and it's difficult for me. But enjoying mathematics and computer programming and computer science can get you really far in the biological sciences now.
I think it has its counterpart in human biology.
Mm-hmm.
So if you're a biologist with these skills, you're looking at brains and bodies and blood and all sorts of things that we haven't looked at. And so then you look at Beckman and places that do this sort of thing.
Yeah.
So, what's the proportion of men to women in this sort of field?
I don't know the specific numbers, but in the biological sciences, I think it's about half and half right now. I took a class at the university, and we actually aired a bunch of I took a class with Dr. Katie Heath at the University of Illinois called Amplify the Signal, and we actually aired a bunch of pieces we wrote here on WEFT, on Smile Politely. And in this class, we got a very unique opportunity to talk to NSF, which is the National Science Foundation. We got to talk to some of the program officers there about what they're looking for in proposals, and what kinds of broader impacts, are parts of your research that are going to reach the community at large, and how is your research going to impact the rest of society. And you have to write about this when you write up grant proposals, when you try to get money. And so we talked to the NSF program officers about what kinds of things they were looking for, and they really like research that targets underrepresented groups. And according to NSF, women are not considered an underrepresented group anymore, mostly in the biological sciences. I don't know about the other fields. Okay. I imagine it's not nearly as even in computer science and engineering, but
this is probably a rude question in science, but how do fashion a family and how do you come back into this sort of field when you've had a child and
oh gosh, I don't know yet. You'll have to get back to me in 15 to 20 years.
There hasn't been a very good system for letting you go for 5 years and come back.
Yes, there in a lot of academia, there still is not a good system to support faculty members, men and women, who want to start a family. There's not. Still socially, there's not really a good system that set up for that. You could be out of action
for 3 years and your literature has escaped you.
Yeah.
So do you take your computer home with you?
Yeah, I don't know. You'll have to ask me when I have when and if I have a family someday.
Okay. We want to just briefly say that when you were here last time, we were talking about your avocation, which is roller skating, and you came in just a little late today because
I came in late today because I have an injury from a roller derby bout yesterday, and it took me a little longer to get here than I thought it would.
So we have a few moments. I was intrigued by how your organization works. You have people up front that are sort of a barrier and people have to get through.
Yes.
If in a very simple way, can you tell people what the fun is of this sport?
The fun for me of roller derby is that I get to spend a lot of time with really interesting, exciting women in the community, and I get to form really great relationships with women I never would have even met otherwise. And it's also really empowering for me to be able to put so much of myself into a very athletic sport. It's very competitive, and I never really I played softball through like grade school and high school, but I never really considered myself an athlete, and I never really thought I would, but now I do, and it's high action, it's competitive, and I get to meet such amazing women, not only in my league but in other leagues, and it's just a lot of fun.
So what do? You go around in a circle. Just explain to
Yeah.
people what do you do.
Oh,
what is
okay.
the
So
competition?
we have an oval-shaped track that we skate on, and there are 5 players from each team on the track at one time. You have 1 jammer, and you have 4 blockers. And the goal of the jammer, she wears a star on her helmet, is she kind of is like a ball. So every time she passes an opposing player, she gets a point. And so the job of the blockers is to stop that jammer and to help their jammer get through. And so we all line up in our positions where we're supposed to start, and then they blow the whistle, and the first jammer to get through all of those blockers becomes the lead jammer. And she now has complete control over the jam. And she can call it off, and she does that by raising her hands and hitting her hips. And so she has control. So it's getting lead jammers really good. And then on their second pass around the pack, which are the group of blockers, they start scoring points. So that's kind of the gist of how you
And you can bump
play
each
it.
other. And,
Yes, and
you
there
got bumped.
are lots of rules and regulations as to how and when you can hit people, but you are allowed to hit people. Not with elbows or with your fists. Yeah, it's very strategic.
It has to
but-
be with your hips or something.
Yeah,
Like that.
yeah.
So what did you damage?
My knee. I hurt my knee. Kind of a reoccurring injury on my knee.
Okay.
What do the other women in the league and on your team say to you when they find out that you're a scientist studying bees?
Well, we actually have a lot of other scientists in our league. So we have a professor in anthropology, other entomologists. We have lots of scientists in our league. So
Well, she has a bee name.
Yeah.
Her title across her back.
my roller derby name is Pollinator. And
good.
just before
It's just too
the
good.
program ends, I just want to make sure that everyone knows that next week is National Pollinator Week, June 17th through the 23rd. And in addition to the nature walk and the photo workshop, we're also having a bee identification workshop that day and some storytelling at the Pollinatarium. And this is all, if you Google National Pollinator Week in Champaign-Urbana, you can find our website, our Facebook, and you can get all the information there.
But if you don't go online, what's the simplest directions you can give for people to get down there?
So we're having all of our events either at Meadowbrook Park or at our Pollination Museum, the Pollinatarium. And you can't see it from the road, but it's on the corner of Lincoln and Windsor. And we have a marked entrance on Windsor. And you'll drive down a driveway, see a bunch of beehives in the woods, and then you'll see a building covered in a mural of beautiful pollinators, and you'll know that you're there.
Cool.
And I want to say that the coneflowers are at the peak this week.
Mm-hmm.
And they look
We've
wonderful.
got purple coneflowers in our front yard. I can't wait for the goldfinches to find them and come
Oh, yeah.
filing on top of them.
They've
and
gone
everything.
from being vertical to them dropping their petals back to make the cone. So
Oh no, I've just forgotten what I was going to say. If people were listening, they've put 2 and 2 together. Dave can't be both doing his thing next week for the pollen and on the air, but we will find something else to have on the air in this time slot. So feel free to tune in if you're not already going to be at the Pollinatarium.
Thanks for remembering
Or
that.
maybe we can all just go down there. Everyone listening will all just go meet with Dave there instead.
Yeah,
next
that
time.
would be great.
Okay, there we go. That's what we'll do instead. We're way over time. Thanks a bunch. Are you graduating?
Me?
Yes.
Probably in a year
Oh,
and a
good.
half, maybe.
So maybe we can have you back again. It
Yeah.
sounds like you're looking for a job, which means we can't talk with you anymore after this
Yeah.
one. So, but okay. Well, they've officially handed me the CD for the beginning of the next show, so we should probably
Dave Monk, your Prairie Monk, WEFT, Champaign 90.1 on your FM dial. And
Dave on the board
Michelle
and
Dwayne.
yes.
And
Yeah, and again, me again. So thanks a bunch for tuning in, folks. The views and opinions expressed here are those of the speakers and do not necessarily represent those of WEFT, its board of directors, associates, station manager, or Prairie Air Incorporated. And with that, it's time now at 2.5 minutes afternoon to go on to Shruti, Music of India, here on WEFT Champaign.